Hi everyone, and welcome to our video on the evidence used in classification. What we're going to be doing in this video is having a little look at the specification reference 4.2.2c part 2, the evidence that's led to new classification systems such as the 3 domains of life, and we're going to be having a look at biological molecules, genetic evidence, and then the details of those 3 domains with a comparison of kingdom and domain classification systems. First thing then, we need to understand a process that can happen when organisms are evolving. And this is a process we refer to as convergent evolution. So what we have at the bottom are 2 pictures of 2 organisms. So we have a dolphin on the left and the great white shark on the right. Now, if you imagine that you knew nothing about life here on Earth, and we just gave you these 2 images, you would look at them and think, well, they're pretty closely related. Because if you look at them, they've got very similar body shapes, they've got fins, they live in water, etc. So they've got a lot of these features that we can see that suggest that they're actually very similar, and therefore you might think they're closely related. But they're not. Hopefully we do know that dolphins are mammals, whereas obviously the great white shark is a fish. So quite clearly they're very different in terms of how they've evolved, in terms of they were not closely related. But why do they look similar? Because of this process of convergent evolution is the answer. When we're referring to convergent evolution, this is where we've got 2 unrelated species that have just adapted in similar ways to their environment. And so even though they are not closely related, they look really similar to one another. So what we find is this throws up a problem for us when it comes to classification, because any of these organisms that have undergone convergent evolution, because of all those observable features that are really similar to one another, then we may want to put them in the same taxonomic group, just based on those features we can see. And obviously this is something that pre-modern technology we were a little bit stuck with, to be honest, because we didn't have the knowledge, we didn't have the technology to be able to gain this greater understanding to actually split them off fully. However, as technology and understanding advances, we have got this, and therefore we've been able to come up with a much better system of classifying. So when we're now looking to classify organisms as being in the same groups as one another, we tend to use more of these modern techniques, which are looking at evidence from biological molecules to really drill down to work out how closely related those species are to one another. Now, when we're doing this, what we need to do first of all is pick a biological molecule that's found in all living things. If we want to compare a diverse range of species, it wouldn't be very smart to pick a molecule that's only in a couple of them. So we pick biological molecules that are found in all living things. So generally these are the ones that are involved in some of those real fundamental life processes, things like respiration, etc. And therefore we can make this assumption that the earliest living things here on planet Earth had identical versions of all of these molecules. And there's a couple of good ones to remember here. We've got cytochrome C and we've got RNA polymerase. So 2 of these key biological molecules in fundamental life processes that are found in all living things. Then, because we've made this assumption that these once were all identical in those earliest forms of living things, what we can now say by looking at those molecules in all the different species alive today, we can look to see how similar or how different they actually are to one another, and then we can work out how far removed each organism is from the next. And we can say that all of these differences we can see in the structure of cytochrome C, the RNA polymerase, are as the result of evolution. When we're looking at these biological molecules, we are literally looking for how similar or how different they are from one another. If we've got two of those biological molecules, so two cytochrome Cs from different organisms, and they're really similar to one another in their structure, then what we can say is that those two species are more closely related and they diverged from one another at a recent time in the past. If, however, we've got quite large differences between those cytochrome C molecules, then we would be able to say these are less closely related and they must have diverged from one another at a much greater time in the past, so much further back in time. I've mentioned cytochrome C quite a lot because I'm trying to get it into your heads, as this is a great biological molecule to mention on your A-level papers. Cytochrome C is one of those proteins that's used in respiration. So every living thing that respires has a version of cytochrome C present. Now, because cytochrome C is a protein, that tells us it's made of amino acids. And what we can do is, just by looking at the sequence of the amino acids within that cytochrome C structure, we can then work out how closely related species are to one another. The more differences there are in that amino acid sequence, the less closely related the species will be. The second molecule we can use for this is DNA. Now, DNA has this universal code, so this means that in all living things, DNA is DNA. It doesn't change. And therefore, what we're looking at is that the same sequence of DNA will code for the same amino acid sequence. So what we can actually do there is, by having a look to see how similar the DNA structures are, again, we can say how similar those organisms are. Hopefully we do remember that we get these things called mutations taking place inside our DNA. And a mutation, in the simplest terms, is a change in the base sequence of the DNA itself. Now, these mutations will happen randomly, and therefore what we do know is that over time mutations will have occurred in that DNA sequence. So because we're looking at obviously something that's happening over a period of time, the more similar the DNA sequence, i.e. the fewer mutations that have occurred between the two, the more closely related those species will be, because they were obviously sharing that common ancestor at a more recent time in the past. Therefore, that DNA sequence hasn't had the chance to undergo so many mutations, so it's still quite similar. We now come on to this idea of the three-domain classification. And in the scheme of classification, this is like modern times. It was back in just 1990. And we have this guy called Carl Woese. Now, he came up with this thing called the three-domain classification model, and he came up with this based on studying the ribosomal RNA genes. Now, when we look at this three-domain classification, what we end up with are these three groups at the bottom. We've got the bacteria, the archaea, and the eukarya. So those are our three domains that we've split everything into. So what he actually worked out from studying this ribosomal RNA and why he came up with these three different domains was, when he was studying bacteria, he noticed that there are some key structural differences in them. So some of them have this very different cell membrane structure. They've got a different RNA polymerase. They don't have proteins bound to the genetic material, and the internal structure of their flagella is different. He then worked out that the archaea and the eukaryotes share certain features in common with one another. So he said that there are similar RNA polymerases, they've got some proteins bound to their DNA, and they've got similar DNA replication and RNA synthesis mechanisms. So when he had all of this information in front of him about, obviously, these features about the bacteria, the similarities between the archaea and the eukaryotes, then he actually said that the archaea, these ones here, are actually more similar and therefore more closely related to the eukarya than they are to bacteria. So even though the archaea are forms of bacteria, then they're not the closest related to them. Instead, what he said was our eukarya, which are obviously our animals, our plants, our fungi, etc., have more features in common with archaea than the archaea have in common with the bacteria. So this is what led to these three domains. So we've got the eukarya, the archaea, which are a lot of the extremophiles, so the ones that live in deep sea vents, etc. And then we've got the other bacteria, so the bits, you know, that cause food poisoning, etc. And this is what we have as our three-domain model. So it was literally looking at those similarities and differences between these organisms, that because we have more similarities between the eukarya and the archaea, then we have a common ancestor here, which is more recent for our eukarya and our archaea than it is for the archaea and the bacteria, which happened further in the past, further down that phylogenetic tree. As always, I do suggest that you subscribe to the channel so you can see when the next video is uploaded, and of course head on over to the A-Level Biology website, where you can find a whole range of different resources, all designed to help you with your A-Level biology studies.